Hello friends, welcome to my channel Clinical Biochemistry by Dr. P.K. Prabhakar. In the series of variants of PCR, this is the third lecture where we are going to discuss about the Hotstart PCR. So we will see what is exactly Hotstart PCR and why we require this one, and then later on we will see what is the advantages and disadvantages of this Hotstart PCR. Literal meaning of this Hotstart PCR is it is a modified form of conventional polymerase chain reaction or PCR, which reduces the presence of undesired product, unwanted extension of the DNA. So all the undesired, unwanted products, the PCR amplified product, that is going to be reduced by this method. Not only that one, primer, dimer, or due to non-specific DNA amplification at room temperature, that is also going to be reduced. So later on we will see detail about that one. So this is the basic conventional PCR where three different stages we are having: denaturation, annealing, and extension. And all three reactions, all three processes occurs at different temperature. Denaturation occurs at roughly 94 degrees centigrade. Then we are having annealing, where primer is going to bind with the single stranded denatured DNA and around 54 degrees centigrade. And lastly we are having extension, where thermostable DNA polymerase extends it. And this is the same process, same conventional PCR in the detailed stage. So this is the first stage where denaturation will be there, roughly 94-95 degrees centigrade, where double stranded DNA will be denatured into two different strands. In the second stage, this denatured DNA is going to bind with the primer. So we will have one pair of primer, that is forward primer and the reverse primer. So this primer binds with the corresponding denatured DNA. And then in the third stage, the thermostable DNA polymerase, which can be preferably Taq polymerase. Other than that one, we are having other polymerases also, like Wnt polymerase, PFU polymerase, or PWO polymerase, or deoxyribonucleotides. This primer will be extended by this thermostable DNA polymerase, and in the end we are going to get two new form of DNA. We started with the one single double stranded DNA. As a result, we have got two newly formed DNA. So this is one cycle. So one cycle of PCR contains three stages, that is denaturation, annealing, and extension. So this process, this cycle is repeated roughly 20 to 40 times for the sufficient amount of DNA formations. Now come to this Hotstart PCR. What is this Hotstart PCR is? The specificity. Normally whenever you are doing PCR, so the specificity is the main aim for any PCR reaction. Whenever you are going to perform a PCR reactions, the main purpose is the specific desired DNA will be amplified. Every PCR modifications, whatever conventional PCR we are going to modify, so we have seen a few of the modified forms of our variants of PCR. Later on we will see some other number of variants of PCR. So any modification in the PCR is to increase their specificity as well as the sensitivity of the reaction. Means only the desired DNA will be extended. So that is the purpose behind any PCR variants or any PCR modifications. Normally whenever we are doing a normal conventional PCR, there non-specific binding of primer to the DNA is the major problem. That is going to extend the undesired DNA. Like this is my DNA, this is my target DNA. Normally we want this portion of DNA to be extended, but if this primer will bind at outside the target, then this portion of DNA will also be amplified. So that is called a non-specific binding, and that is the major problem in any PCR reaction. The non-specific binding increases the chance of false positive result. So we will have false result, we will have negative result. In the case of Hotstart DNA, which is the modification of the conventional PCR, it is going to reduce the non-specific binding by reducing or by limiting, by restricting one of the component, one of the reagent, until the heating step of the PCR will be processed. Means we have seen three different stages. Till the heating process will be completed, one of the component will be inactivated or non-functional, so that polymerization will not initiated or extension will not started. So by that method this Hotstart PCR works. So primer, dimer or mispaired or false prime the target are the major region for the non-specific binding, and successful amplification is the main goal for amplification reactions. So however, non-specific binding is the major barrier in this PCR reactions. Whenever primer will bind at the non-specific region, it will extend the undesired DNA, and when undesired DNA will be extended, you will get the wrong result. Why this non-specific binding occurs? What is the reason behind that one? So there are 4 specific reasons for that one. The first is the annealing temperature. If the annealing temperature is the temperature where primer binds with the DNA, means separated DNA. So in the case of annealing temperature, if the annealing temperature of the reaction is lower than the original annealing temperature, or means initially it was a certain temperature, and if by fortunately or unfortunately if annealing temperature has been reduced, so decrease in the annealing temperature results in the non-specific binding of the primer, and that will give you wrong result. So decrease in annealing temperature gives you non-specific binding. Second one is specificity of the primer. Now if the primer binds at multiple locations in the genomes or the target DNA, that may amplify more than one fragment of the template DNA, and it results in more than one bands in your gel electrophoresis. So primer should be very specific, and it should bind to the target DNA only. Third is the higher concentration of PCR additive. The PCR additive plays a very critical role or crucial role in the achievement of amplification of those templates also, which is normally impossible. But the higher concentration of any of these factors, additives like divalent catalyins, magnesium chloride or potassium chloride or DMSO or other facilities, it increases the non-specific binding. So more concentration of these additives, more non-specific binding will be there. And the fourth is early action of Taq DNA polymerase. Taq DNA polymerase is a thermostable DNA polymerase, which normally going to elongate the primer binding. Once primer will bind, it will elongate the primer. So the reaction preparation time is a major factor that induces non-specific binding. At the room temperature, normal temperature also, the Taq DNA polymerase actively involves in non-specific amplification. So these are the 4 major reasons why non-specific binding, non-specific extension occurs. Okay. So the reason for this is the early reactivity of the Taq DNA polymerase. All the components such as deoxyribonucleotides, template DNA, PCR additives and primers are there in the PCR tubes before putting in the PCR machines, because you are having a tube. In that tubes you have everything means you are having template DNA also, you are having primer also, you are having nucleotides also, you are having buffer also, you have all these components also. Okay. The Taq actively starts the synthesis of the DNA by adding deoxyribonucleotide randomly. Due to the non-specific binding, the yield of the original DNA results as well as our target amplificons is going to be decreased. So that is a drawback of this process. So if you will see here, this is the conventional PCR where we are going to have normal means pen drops, and we are going to have all the components, and then we are going to put on the PCR machines, and then it will be going to give you the PCR products. In that case, we are having PCR products also and non-specific binding also. Then if you see in case of Hotstart PCR, we are going to, this is here you are going to see one of the component, that is the wax. So this wax is normally going to inhibit, or it is going to protect, works as a barrier, which does not allow DNA polymerase, Taq DNA polymerase to come in the contact of the DNA template. So extension will not be there. Once this, because of the heat, once we are going to heating the temperature at 94-95 degrees centigrade, this wax will be melted, and then this DNA polymerase will come in the contact of the template DNA. In that case, you don't have the non-specific binding. So the difference between this result and this result is only PCR product is there, non-specific binding. So this is one method. We have many methods by which we can do this kind of, we can achieve this process. So we will see. So exactly what is hot start PCR? The hot start PCR means it is equal to one of the component starts its activity under the hot condition of PCR. Means because of the higher temperature, which component we have inactivated, that is going to start after the heating process only. So the hot start PCR is the most advanced modification of the conventional PCR, in which one of the PCR reagent is activated only after heating. Till heating will be there, PCR process will not start. So the non-specific binding and primer dimer decreases the yield of the reactions, and our DNA of interest, or gene of interest or template DNA is amplified very less. So the aim of the hot start PCR is to limit the reaction at an early stage before heating process. So normally the PCR process which should occur after heating process, because of the Taq DNA polymerase, it starts before heating. So this hot start PCR normally decreasing those process, the early starting of the elongation process. So by limiting the Taq DNA polymerase in the reactions. This is the now conventional PCR, which just we have seen it. Every component is there. So in that case, DNA polymerase is also there, nucleotides are there, templates are there, means buffer are there, everything is there. So this Taq DNA polymerase or DNA polymerase actively polymerizes the DNA before heating process also. So non-specific DNA also and the PCR product also, both will be there. But in case of hot start DNA, we are going to DNA polymerase will be inactivated or separated from the DNA template. So because of that one, this polymerase cannot polymerize the reaction till this heating process will be there. So once this heating process will be there, then DNA polymerase becomes active and extension will be there, elongation will be there, or PCR process will start. So in that case only PCR product you will get it. So non-specific extension or elongation of the DNA will be stopped here. So that is the advantage of this hot start PCR. There are different ways, there are several ways by which we can avoid the non-specific bindings. So you can overall divide in two different ways. First, inactivation or inhibition of Taq DNA polymerase, where Taq DNA polymerase will be inactivated or inhibited. Or second one is we are going to add DNA polymerase after certain processes. Okay, so in the case of first, where DNA polymerase will be inactivated or inhibited, either you can use enzyme-linked antibody or high specific oligonucleotide can be added, or wax barrier can be used. Or in case of when we are going to use Taq DNA polymerase late on, there we are having preheating, freezing, or late addition of Taq polymerase. Overall, either by this process or this process, you can perform hot start PCR, and then the ultimate product is you don't have non-specific binding, non-specific extension will not be there. So only the PCR product will be there. So now we are going to see all these 6 different stages one by one. So the first, when we are going to use by using enzyme-linked antibodies. This is a very novel way to inactivate Taq DNA polymerase. Here the temperature sensitive antibodies, so we are going to take antibodies that is the temperature sensitive. So the polymerase is linked with the specific antibody, which makes it very busy, hence it cannot amplify the DNA early before the reactions. So temperature sensitive antibody inactivates the DNA polymerase because antibody binds with DNA polymerase and it cannot amplify the DNA before the reactions. Once temperature reaches up to the more than 70 or 80 or 90 at a specific temperature, so above 70, once it is going to reach, this antibody, which is a protein component, this is going to be inactivated. So antibody is degraded, and once antibody will be degraded, Taq DNA polymerase becomes free, and then it is going to perform the complete reactions. So in this case you can see at a normal temperature this antibody is going to bind with the enzymes Taq polymerase, so it is inactivating these enzymes. Once the heating process will be there, this protein will be, this antibody will be degraded and our DNA polymerase becomes free. As DNA polymerase becomes free, it becomes active, and then polymerization will take place. So overall, the limiting of the antibody linked, the limitation of this process is, every process is having certain limitations, certain advantages. So when we are going to use enzyme linked antibody, that have certain limitations. What is the limitation? The limitation of this antibody linked enzyme method is the cost, because you need a monoclonal antibody type things, and it will be very, very costly. So the use of antibody increases the overall cost of the reactions, and every time the different enzyme you are going to use, you require a different type of antibody. So that makes this process very costly. Second one is preheating the PCR machine. In this case, in this method, heat the PCR machine at 95 degrees centigrade before mixing all your components. So machine will be preheated at 95. Then we are going to prepare all the reactions on the ice at 4 degree and immediately put the tube into the PCR machines. Means you are not going to give sufficient time to the reaction mixtures before 95. So at a normal temperature, and this is going to decrease the chances of Taq DNA activations, because Taq polymerase normally works more preferably, or optimum temperature will be above 70. This also, the mispaired primer are released immediately. So that is the second one. Third one is freezing the PCR reactions. So this is another method, is deep freezing the PCR mixture. Once the DNTP deoxyribonucleotides, primer, water or templates are added into the reactions, like this is a tube where we have added nucleotides, primer, mix buffer and template, then this is going to be frozen. So these things will be, reaction mixture is frozen. After that one, Taq DNA polymerase and PCR additives such as magnesium chloride or any other divalent metallions will be put it on the frozen surface. Because this is frozen, so this enzymes and PCR components, these components will not come into the contact of DNA. And as soon as temperature will reach more than 70, as soon as this frozen process will be over, means this freezing means frozen things will be removed. So that is going, then these enzymes will come in the contact of template and polymerization will there. So by doing this one also, non-specific binding will be avoided. The fourth important step is, fourth important process is addition of Taq DNA polymerase separately. Once the reaction is prepared, it is placed into the PCR machines, and when the temperature is achieved at 95 degrees centigrade, then we are going to add the enzymes. So in the tubes you will have everything except Taq DNA polymerase. Once this tube will reach up to 95 degree or 94 degrees centigrade, then we are going to open the tube and put it this Taq DNA polymerase. But in this method is not very reliable, because if you are going to open the tube, it may get cross contamination or reaction failure may occurs. So that is the drawback of this stage, this process. Fifth stage is using the wax, which we have seen in the picture also previously. This is very interesting, where temperature dependent wax beads creates a barrier between the Taq DNA polymerase and other components. The DNTP, water, primer and template DNA are added into the bottom of the tubes. Then we will have wax bead. So wax bead will be between the enzyme and other component. As soon as this enzyme, this component, then we are going to put the enzymes and magnesium chloride. And when we are going to heat these things more than above 70 degree, the wax will be melted, and then DNA polymerase will come in the contact of the reaction mixture, and amplification process will take place, as you are going to see here. So this is the wax, and enzyme will be set above this one. This wax will work as a barrier between these components and these enzymes. Once we are going to heat it, this wax will be melted and enzymes will come in the contact of reaction mixture, and ultimately extension will be there, polymerization will be there. And the last process is, we are going to use high specific oligonucleotides. In this case, below the temperature of 50 degree, the Taq DNA polymerase remains inactive in the presence of high specific oligonucleotides. After the temperature above 50, means above 50 degree, the oligonucleotides are detached from the Taq, and Taq releases it into the reactions. These are some of the best methods used for the Hotstart PCR. Okay. Nevertheless, the best adapted method for the Hotstart PCR is an enzyme-linked antibody, wax bead, or a specific oligonucleotide. So out of these 6 three best processes are either you can use enzyme-linked antibody, which is very costly, then wax bead, and third one will be a specific oligonucleotide component usage. This is the roughly process of how Hotstart PCR and why Hotstart PCR we require. Okay. In the amplification of low base pair amplification such as 80 base or 90 base, due to the early amplification non-specific, that is binding, the chances of false positive results are very high. Because of the presence of Taq, the primer can bind to the 4 to 5 specific bases and produce a short stretches of the small base pairs. This process, one of the amazing use of this Hotstart PCR is in the identification or detection of the diseases, and that is TB PCR. We can call it TB PCR. So later on I will talk about this TB PCR also. In the TB PCR, the chances of the infection is very high while we are performing the DNA extraction. Hotstart PCR helps to overcome this problem. This sample can be directly be added into the PCR tubes along with all the reagents. The first step of PCR is extended up to 10 minutes for 94. The bacterial cell membrane is degraded and the DNA comes out from the bacterial cells, immediately followed by the annealing step, and available DNA template will be amplified. This method is very rapid, safe and accurate. Now come to see the advantage. What is the advantage of this Hotstart PCR? The first advantage is it is going to reduce or decrease the non-specific binding. Because it is going to reduce the non-specific binding, so obviously non-specific amplification is also going to be reduced. Also, it prevents the mispairing, mispriming or primer dimer formations. By forming Hotstart Taq DNA polymerase, the reaction can even be prepared at room temperature. And last one is it increases the yield and accuracy of the results. So these are the advantages. This is most important advantages. This is the second most important advantages of this process. There are certain disadvantages also there. What is these disadvantages? The overall cost of the reaction is going to be increased due to the use of antibody or the wax bead. This will be the wax bead. Another disadvantage is that it cannot amplify the larger fragment of DNA. It is applicable only for the small stretches, the small fragment of the DNA. And the heating step is predominant in the Hotstart PCR. Hence, due to the higher temperature of a longer time, the template DNA can damage or break down badly. Then something about the Hotstart Taq DNA polymerase, which I have told you. The important player of the Hotstart PCR process is the Hotstart Taq DNA polymerase. The Hotstart Taq DNA polymerase is different compared to the normal Taq DNA polymerase. So we are in case of Hotstart Taq DNA polymerase, we are going to use this enzyme, which is different from this normal Taq DNA polymerase. The Hotstart Taq DNA polymerase are either enzyme-linked, or it will be oligonucleotide-linked, or it will be chemically modified inactive enzymes. This facilitates the decreasing in the non-specific binding and primer dimer formation. So from the 20 unit, normally it is present in the 200 unit stocks. We can take 2 units of Hotstart Taq, and that is sufficient for 30 microliter of the reaction. The commercial kit, when we are going to purchase it, that contains 10x reaction buffer with MgCl2, KCl and other additives. You can use 1x buffer also. And this overall advantage of this process is it facilitates the specificity, decreasing the non-specific binding and primer dimer formation, non-specific amplifications. Lastly, if you will see the conclusion, the Hotstart PCR is one of the best variants of the conventional PCR method, which gives you best and accurate result. The overall idea of developing the Hotstart PCR is to improve the performance of the reactions, to reduce the non-specific amplification, non-specific binding of the primer, and the technique is one of the best choice to diagnose the inherited disease. So this is roughly all about the Hotstart PCR. Next day we will come with another variant of the PCR. So if you have not subscribed my channel, if you are new to the channel, you can subscribe. You can press the bell button so that you will get all the notification on time. If you have any query, any comments related to this topic or related to any other topic, you can write in the chat box, you can write in the comment box. Certainly I will give you answer. Thank you very much. Have a nice day.